Okay, well, first of all, thank you very much, Doron, and all the organizers of this very nice workshop. Very happy to be here. So I realize my talk really gets along very well with the two previous ones. I'm also going to talk about BCI, learning, and peace-oriented, actually. I'm working in a neuroscience center, so it will be quite neuroscience grounded. And my point, I guess, is to, in complement to Fabien, actually, is to could be summarized by the fact that we can probably take more information than we do so far from EEG data to further improve BCI. So let's see how maybe we can do that. I don't have to go through this because Ori did it nicely. But I'll take this example quite all along the talk. But basically, it's a good example to emphasize that with BCI, we usually reason that we look for discrete mental states to classify, right? In this example, it's a much simple case. We need to disentangle between two states, basically, target or non-target stimulus, thanks to this P300 response. But of course, this is what the book will display. This is far from the real data. Brain responses are considered to be binary, but probably they are not. We know they are not. And although in the methods we use, we consider the variability, this variability usually is considered as noise. Okay, we can identify factors behind this noise. They can be external, basically artifacts, or internal, human factors. But how do we deal with this? Or do we try really hard to understand it, or maybe to use them? So the question, the fact is that modulations of responses are quite often overlooked. And of course, this modulation, they can be between subjects, right? And they can be very useful. I'll take here another example of a node ball paradigm. So the peace-oriented is a node ball paradigm because you have a rare, interesting stimulus, which is target. Here, this is another famous paradigm that you might know, which is the auditorial ball paradigm, where you don't have target and non-target, but what you have is a standard tone, which is often repeated, like beep, 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 but suddenly a deviant, rare tone, bop, which make a surprise. But this can be a passive listening that you can use in coma patients, for instance, to probe their perception. And what you observe when you average the responses is a response to standard tones and here to deviant tones. And the difference between the two is a mismatch response called the mismatch negativity, sometimes followed by the P3 component as well. And this is actually used in clinical routine in our city. First of all, because it has been demonstrated that absence of early components like this one is a good marker of poor coma outcome. But my colleagues have more recently demonstrated that the presence of these late components, more cognitive components, are good marker of good coma outcome, right? So this difference between subjects is very relevant and very informative. But can we go beyond this, again, binary information and average information over a lot of trials or even patients or people? Okay, to talk about factors that are nicely represented in EEG data, I'll take another example of our data, actually BCI P300 experiment, where we also looked at another type of mismatch response, which is called the feedback-related negativity. In fact, it comes when you display the outcome or the feedback of the spelling. So here is the average response over many subjects, where you can see what happens in red when there is an error, a bad spelling, and in green when there is a correct spelling. And you can see a mismatch response, a bit like the MMN, roughly at the same latency actually, and also followed by P300. Well, in this experiment, we tricked the subject. Actually, we used a fake feedback. And we used it in a way where we played with the expectation. So implicitly, the subject could learn that some feedbacks were more predictable than others. When you do this and you compute the difference between those two responses, so the mismatch response, and you compare the two experimental conditions that we had, quite expected situations and unexpected situations, you can see that the fact that there is a surprise in the unexpected case enhances the signal. So this difference, this mismatch is higher with less predictable situations. So these ERPs, they display, basically, they reflect the surprise of the subject, right? In this experiment, we were also monitoring the true feedback, the true attentional effort, which was either producing a P300 or not by the subject. So we could look at the two other conditions, which were actually related to attention, true attentional efforts from the subject, by comparing this mismatch response, whether there was a hidden real error or real correct signal from the subject. And again, we could see a significant difference, which was telling us that attention yields a larger evoked response. So 2 factors here, surprise and attention, do actually modulate evoked responses. So the partial conclusion is that, evoked responses are often considered as binary in BCI. It's already very useful even for the clinic. But however, we know and we showed that a lot of factors, expectation, attention, motivation as well, influence those responses, and maybe we could use that. The question is now, so far I showed you an example where still we average the data over trials or even over subjects. Could we make sense of modulation for trial to trial now, really at the single trial level? Well, for instance, do evoked responses reflect learning by the subject? A bit like what motivated Ori to use LSTM, right? The influence of time. So I'll show you briefly a study where we tried to exploit this. And so this study is about implicit learning during an auditorial task. And we wanted to be very precise, so we used what we have the chance to have in Lyon, which is an MEG system, which we combine with EEG. And our grounded hypothesis was based on the predictive coding or Bayesian brain framework you might have heard about, very trendy now. And from this framework, we derived the following questions or hypotheses. First of all, we thought, okay, evoked responses are modulated by surprise. They should be somehow reflect learning, including automated or implicit learning. Second, we said this learning process has an aim, which is to optimize prediction of incoming events. So basically, in the oddball paradigm, what has to be predicted is when this deviant sound will occur. And then we thought, okay, let's be more precise in a computational way. We think that evoked responses reflect prediction errors, so surprise per se, and more precisely, confidence-weighted or precision-weighted prediction errors. So we could maybe design computational quantitative models to try to fit the EEG data at the single trial level. Right. So in a way, this is the update we were considering that the brain will implement is that the belief will be updated from prior to posterior by a term, an update term, learning term, which would be this precision-weighted prediction error. Okay, here is the design we used with 20 subjects in the EEG and MEG machine. It was implicit because they were watching a movie, so we checked that they didn't pay attention to the sound up to the point they would catch the differences in sequences. And we used 2 different conditions, basically a predictable sequence, where you can see the number of standards preceding a deviant was increasing by a step of 1. At the end of the cycle, we were starting again. And in the unpredictable sequences, the same kind of chunks were simply randomized. Okay, we wanted to be very fine in our extraction of EEG data. So what we did, thanks to the coupling of EEG and MEG, is to do source reconstruction to infer the EEG data at the cortical level. But first, we tested what we observed prior with the FRN, which is, okay, on average, do we see an effect on the mismatch of the predictability? And indeed, again, you can see that predictability yields a reduced surprise response. Okay? So we were quite confident that this will enable us to show some learning trace in the single trial data. So we applied source reconstruction as a fine spatial filters, combining EEG and MEG data to reconstruct the network, cortical network behind this response. We could see actually quite an expected network with activation of Herschel gyrus, more anteriorly the planum polare, and some frontal regions on both sides. And we use single trial reconstructed responses for each subject in those regions to test our hypothesis. Now, what kind of hypotheses did we test? Well, we considered 3 simple alternative models. One is a dummy null model stating that EEG in black here is simply noise or 0 all over, so the brain is not perceiving, right? Then we consider a classical non-learning situation, traditionally in the head of people doing experiments with the MMN. It states that, okay, the MMN, this mismatch response, is on and off. Every time you have a deviant, you have a bit, an amplitude. And our third and preferred model was stating that there is some learning going on with some constant parameter, tau, and this makes your response dependent on the history of the sequence, basically learning, right? Okay, we confronted these models to the data, but remember, we want to fit this to single trial data. So here you have trials, and here you have peristimulus responses. So somewhere you have a 0 where the stimulus comes on, and then this unfolds. So when we fit this at a given latency, and we do that for all latencies like this over trials. Okay, this is the results quickly of our 3 hypotheses. So those graphs tell you for all the 6 regions of these networks when and if one model was the best explaining models. And what you can see is prior to stimulus onset, which should be at 0 here, the null model is the best explaining model. But at some point, and the story is pretty much the same all over the network, at some point the learning model prevails, and this actually happens at the latency of this mismatch negativity that you can see on averaging, when averaging your data here. Okay, so this learning model was prevailing here, telling you that you should not average. Basically, you have more information than we thought in these signals. And I told you the learning has a parameter which is this tau, and this tau indexes the memory of the learning. So we compare now tau between the 2 conditions we had, predictable sequences and unpredictable sequences. And what we observed at the group level is that in the predictable sequences, this tau increases. And this makes sense. It means that perception of a predictable sequence is associated with a larger memory of past events, right? And, sorry, this comes later. It makes sense because if you have information in the sequence, and basically you have when it's predictable, well, it means that intuitively you should increase your memory because this information is useful to better predict. If the sequence is randomized, you don't need that information. So you can reduce your window, right? But we had a final prediction based on this observation, which was at the computational level. This means that when you can predict in predictable sequences, you should have less prediction error on one side. But you also should have a higher confidence, higher precision of your prediction, because you have more information you can actually predict. Okay? So these predictive coding-derived predictions can actually be tested using now models of effective connectivity of the electrophysiological data, because predictive coding tells you that those quantities, like prediction error and precision, they are actually coded in the network. And the way they are coded is as follows. So what we use to test this is a framework called dynamic causal modeling, which tries to explain EEG and MEG data by not only a spatial network, but also a temporal coordination, an organization of this network, a hierarchical organization, starting from input regions and propagating with forward and backward connections. So we found this network for our data, and then we tested specifically the hypothesis derived from our computational analysis. And what we found is that the forward connection are actually upregulated in the unpredictable condition. Meaning, and this is very well with the idea that the prediction error is higher in unpredictable condition. Right? And actually, interestingly, we could see this. There is a trend with MEG, but it was really salient in EEG. And for the second prediction, we could see that intrinsic inhibition, which are associated with reduced precision weight or reduced gain, it's a bit like implicit attentional gain, if you like, is downregulated in the predictable condition. And interestingly, I don't know why, but this was really salient with MEG and less with EEG. Right, so to conclude, we saw that, I hope I convinced you, although quickly, that troy-wise modulation of evoked responses are not just random noise. They do reflect implicit learning and adaptive mechanism, like in the experiments I showed you. To show this, we used at least 2 types of computational models. Models of mental operations, of learning, right? But also models of the genesis of neurophysiological mechanisms and responses. So those models, they enable us to embody generative hypotheses about our data, and they inform us about what Evoked responses do represent. So my last word will be about what we do now to kind of exploit this knowledge or go further and to the application world. So what we have two of large projects related to the P300. One is investigating the variability of Evoked responses in patients, so locked-in patients, because you might know that P300 splitter does not work as well as it should do with patients, and we'd like to understand why. Another line of research is also to use actually P300-based BCI, or games actually, to train children with ADHD in a kind of neurofeedback training way. So we have actually a clinical study running right now using this to train attention. And I think I'm done. Yes, I'm done. Anyway, thank you.